There we go. Okay. Yeah, so it's related to, that's a way of saying that I don't actually work on autism spectrum disorder, but I got invited because, I guess because the people here thought that it might be relevant, so I'll try to make it that way. There are several points that I really want to make out of this talk, some of them scientific, some of them maybe scientific political, and as the last speaker, I get to be as outrageous as possible, perhaps. But I'd like to address, in a global sense, why is there such an increase in neurodevelopmental disorders, including ASD? Now, I'm one of those people who thinks you might be barking up the wrong tree to focus solely on autism, because in fact, autism is just one of many neurodevelopmental disorders. And just as in the field of cancer, you can learn a lot about breast cancer by studying cancer in general, I would submit to you that you can learn a lot about potential contributions to autism spectrum disorder simply by regarding all of neurodevelopmental disorders as society's problem. I think this one, it's almost a no-brainer, but I'll brain it anyway. Why do neuroactive agents in particular produce permanent alterations with developmental exposures? You know, you use psychoactive drugs all the time, right? Whether you use alcohol, right? Does that change your behavior permanently? No, but alcohol exposure during development changes things permanently. So what is it about development? What is it about the brain's plasticity that makes it do things that it thinks are right for the time of the exposure but turn out not to be right later on? Why is there a critical period for these effects? Why is it that exposure during this period does all kinds of weird stuff and the next period it does a whole different set of weird things and you get older and you just become degenerate, right? Why are there critical periods for that? This is almost like anti-intellectual, because in basic science it's all about mechanism, right? That's all we care about. Why do apparently unrelated agents produce the same outcome? Does it even matter what the compound is so long as bad stuff happens in the critical period? And I'm going to try to develop the theme that in some sense mechanism is relatively unimportant in looking at neurodevelopmental disorders, and what might be more important is convergence of different kinds of mechanisms on final common pathways and targets, so that two things that work by totally different mechanisms nevertheless can give you the same adverse outcome. And I'm going to give you examples from one class, from environmental chemicals, organophosphate pesticides, and those have already been mentioned before, and examples from a prenatal drug exposure, terbutaline. Terbutaline's used incorrectly to arrest preterm labor. And I'll show you how those two converge on the same kinds of things. So here's a set of facts, okay? The rest of it will be like the usual bullshit of overinterpreting the science. But this is true. 5,000 new chemicals appear on the market every year. I spent 10 years getting chlorpyrifos out of contributing to major research that got chlorpyrifos out of the house. In that period, Dow Agro produced 6 new organophosphates, all right? There's 5,000 new chemicals appearing each year. EPA estimates that probably a quarter of them are neurotoxic, all right? 2/3 of the high production chemicals, the ones in thousands of tons, never get tested for neurotoxicity. Chemicals aren't drugs. Drugs have to jump through some hoops. Chemicals, we get a rat LD50, we do pathology, we go into production and we see who gets sick, right? We're the test animals. We know that the developing brain is more vulnerable to these chemicals than the adult brain, so God knows what the hell's going on with all these neurotoxic chemicals. We're awash in. And here, this is where facts, and you can draw the connections between those things and the stuff down here. 17% of US school children now diagnosed as suffering with neurobehavioral disabilities at an annual cost of between $80 and $170 billion a year, okay? It's less than the Iraq War, but it's a hell of a lot more than we're getting in science, isn't it? 250% increase in ADHD diagnosis in a decade. 190% increase in children requiring special ed for learning disabilities in a 2-decade period. And I don't have to tell you guys about the increase in diagnosis of autism spectrum disorders. So I stole some slides from people who go before Congress because I'm too chicken to do that. No, I'm too basic science to do that. I'm one of these people who only is interested in humans if they're good models of rodents. So they don't want me speaking before the scientists. So this is from the LDDI initiative, which was published just a few months ago. what we know about chemicals as developmental neurotoxicants, and what they said is there's 10 compounds that we know for sure. We know how they work, their mechanism, the sensitive periods, and think about it. We don't even know how lead affects the developing brain. We know what the outcomes are, right? But we don't know the mechanism, right? We've got about 200 where we have good evidence. Lead falls into that category. 1,000 suspected. I would put the organophosphates into the good evidence. Dow Agro would put them into suspected or unknown. And more than 80,000 chemicals in production whose toxicity we know absolutely nothing about. And you're being bathed in them, and your kids are being bathed in them, and pregnant women are being bathed in them every day. So Phil Landrigan and Grandjean published a little diagram, which I think is very illustrative about how we learn about neurotoxicity. Neurotoxicity in adults, and this is reciprocal dose here. So the lower the curve, the higher the dose, and this is the number of people affected. Neurotoxicity in adults, well, Paracelsus described lead poisoning, right? Poisoning incidents, that's how we learned about methylmercury. Methylmercury, you probably don't remember, I'm old enough to remember. Methylmercury was used as an antifungal because it was safer than mercury, okay? It took the poisoning incidents at Minamata Bay in Iraq for people to realize safer for you, but not safer for development, specific developmental neurotoxicant. And then, of course, there are all these other compounds where there are subclinical effects in child populations. And that's really Herb Needleman's lead story about children and lead is subclinical effects. That was the jump, that you didn't actually have to be poisoned in order to have effects. And then here's all the rest of those compounds. The silent pandemic. So, I'll draw the connection. If we want to see where the origin of big rises in neurodevelopmental disorders are, you just check the urine of any school kid today. Compare it if there's still some urine samples back from my childhood, and you'll find a lot of weird stuff in their urine that wasn't there in mine. My mother just smoked during pregnancy, so I was actually supposed to be a football player like Dan Marino, and instead here I am giving a talk. So I'm going to concentrate not on just any old chemical, but on the specific question, why it is that chemicals that have specific activity towards the nervous system, drugs that are neuroactive, or chemicals that are designed because they're neuroactive, and the pesticides are the obvious choices there, why these are so particular, why they all will inevitably be disruptors of the developing brain, produce neurodevelopmental disorders. And part of this has to do with something that we've actually known in some form for nearly 40 years. And that is that if we look at neurotransmitters, those chemicals that in our mature, or my semi-mature brain, communicate things across the synapse. There's, presynaptic terminal releases stuff, it goes across here, and it discharges this one, and I can think, therefore I am, right? Those very same chemicals during development are morphogens. They don't just communicate synaptic function, they communicate from one cell to another how to develop. Right? And it's something that exists because we took it over from lower organisms. Sea urchins assemble their embryos using acetylcholine, norepinephrine, dopamine, serotonin, all those things that are neurotransmitters are morphogens in the sea urchin. So if you're Mother Nature, and if you believe in Darwin, which they're supposed to do even in Tennessee by now, right? Despite the Scopes trial, there's a mechanism for development right there, so we take it over, and that's the way our brains assemble. Well, that's good and it's bad, right? So here's a nerve terminal releasing a neurotransmitter onto a developing target cell. There are neurotransmitter receptors here. It activates signaling cascades. And, it's the same neurotransmitters, same receptors, same signaling cascades that make a mature nerve cell communicate across a synapse. But the difference is during development, these same signals, same receptors, same signaling cascades evoke a change of the repertoire of genes that are being transcribed. And the repertoire that you read out depends on what the developmental state of the cell is. So at one stage, early on, as we were just hearing about cell replication, that neurotransmitter signal will tell cells, okay, divide, make some more, right? But slightly later, that very same neurotransmitter, same receptor, same signaling cascade will read out genes that say, stop dividing, differentiate, specialize, be all you can be, right? But one of the things you may want to become if you're a nerve cell is dead, right? Because the brain models itself by killing off, generating and then killing off excess nerve cells to make room for connections. It ain't how many cells you got, right? It's how much you got in the way of connections. Well, all of these signals at different stages are influenced by the milieu of synaptic transmission, by neurotransmitters, right? And that's kind of cool. It means that the brain has a self-assembly program. From a few primitive connections, you have a wave of differentiation. But it also means that it's modifiable. We don't all have the same exact brain. It's modifiable because, well, you know, you stimulate a brain, you get sensory input. Mom wants to listen to Britney Spears, makes you dumb. You know, so certain neurons fire. The fetus learns, okay? And that means that because nerve activity is manipulatable by the sensory environment, the brain becomes individual. All right, now let's, instead of just leaving it in its wonderful state, let's throw in a neuroactive drug. One of the things I won't talk about, but I usually do, is smoking during pregnancy. Mom takes in nicotine, which works because it pretends to be acetylcholine. So you have all this exquisite stuff that's supposed to be timed, like playing Beethoven's Appassionata Sonata, and some clown comes over with a 2x4 and mashes all the keys down while you're simply trying to play, right? Well, the message isn't going to get through. Well, it's all the right signals, it's all the same keys, just not coordinated anymore. And so what happens is some cells will be thrown out of replication too early, some will differentiate into the wrong phenotype, some will die that was supposed to live, some will live that are supposed to die. And the point is that the same neurotransmitter is used for all these decisions. So this tells you why there's a critical period. If you put in an artificial input into that critical period, you're going to change the fate of the cell. And that produces a permanent change in what happens. If you do it after the critical period, you get a response the way our brains do. You know, you walk into the elevator and somebody did something they shouldn't and it smells bad, but if your elevator's stuck for 3 or 4 floors, you stop smelling it. You adapt. That's what our brains do in the mature sense. But the problem is, of course, for development, all right, it isn't like there's a critical period. There's a whole series of critical periods, right? And the outcome that you get depends on when that exposure occurs. And it'll be a different outcome for the same compound in different exposures. It's not like mercury always does this. It's mercury at this stage does that. Mercury at that stage will do something else. Now, how do we get to the same, that's how you get to different outputs from the same compound. How do you get to the same outcome from different compounds? So what I've done here is to schematize screwing up function of a synapse. Here's a normal synapse, presynaptic terminal neurotransmitter, target cell with its color matched so it's got the right receptors. I could damage or lose the input, right? Less neurotransmitter, screwed up connection. I could damage or lose the target. Those are very different mechanisms, aren't they? Completely different cell being screwed up, but the outcome is the same. I could miswire the connection, all right? So this neuron's supposed to go here. It goes to a cell that sort of has some of the right receptors but might not be, but it's connected to something else. Or I can do something that's becoming more and more apparent as one of the main outcomes of developmental exposures to neuroactive agents. I can switch the phenotype so that here's a cell, for example, making acetylcholine, and now it's juxtaposed to a neuron, a target cell that's got serotonin receptors, or vice versa. And I'm going to show you examples of that. So the corollary here is that exposure to all kinds of different agents can actually produce the same effect, or if you've got sequential exposures, 2 different hits, you can get additive or synergistic effects from compounds that are mechanistically unrelated to each other. With that in mind, let's go attack somebody. Right? See, when we did the genetic stuff this morning, you could attack your mother for your genes and your father. We're going to attack the chemical industry instead. They're easier to sue, too. So maybe this information be of use to you. You know, this is going to get me in trouble because it's being recorded, right? Oh, well. She just hit the red, okay, yellow, she thinks. Fair enough. So here's chlorpyrifos. Now, chlorpyrifos is one of the organophosphates. The organophosphates represent half of all the insecticides used worldwide. And despite everyone talking about chlorpyrifos is out of the home, in fact, half of the organophosphates used in the world are still chlorpyrifos. The ban for use in the home ignores the fact that 85% of it's used in agriculture. So if you're working in the San Joaquin Valley or you live there or in any of the agricultural communities, you're still getting crop dusted, okay? It's not an endocrine disruptor. In fact, these compounds were there to replace organochlorines because it turns out DDT is bad for birds but okay for humans, and organophosphates, bad for humans, okay for birds, I guess. As we regulate it, what happens is people dump it into unregulated sites, so it's a disposal problem. And actually, this is why I'm glad I have a Macintosh computer for this one. if you've ever done these things where you go across platforms, it's the symbols and bullets that change. So this one that says nerve gases in warfare, the last time I gave a talk and it went onto a different computer, that turned into a smiley face. People think I'm stranger than I am. So the point about organophosphates, okay, is it turns out the developmental neurotoxicity was totally unrelated to the mechanism by which it poisons adults, which meant that all the regulatory standards were incorrect for protecting the developing fetus and child. And the effects are subtle but widespread. You know, this is true for all of the kinds of things we're talking about. The sneaky developmental neurotoxicants aren't things that blow holes in your brain. We can identify those. It's the ones that are sneaky bad that are the ones that are a problem. And although, you know, I'm going to show you stuff that we modeled in animals, this has now been taken on for that lesser species of humans, and everything that we have found in the animal models is now appearing as studies of inner city and agricultural populations, both in New York and California. And as has been mentioned, developmental exposure to these increases autism risk. So chlorpyrifos, one of its actions is to increase the concentration of acetylcholine in the synapse by inhibiting its breakdown. That's the boring mechanism. Direct actions on receptors, direct action with signaling cascades, changes transcription factor expression function. I left out in this one oxidative stress, causes oxidative stress. It's like the perfect compound. You can inject it into a rodent and it produces a paper, right? So the point is that here's a compound that doesn't have a mechanism. It has a family of mechanisms. So it does all kinds of things. And we're going to focus on one of the critical periods in late gestation to the early neonatal stage, which is equivalent to second trimester in human fetus. And I'm not going to show you every neurotransmitter under the sun. I'm going to concentrate on serotonin. It got mentioned earlier today as one of the potential issues in autism. So chlorpyrifos produces miswiring of serotonergic systems through this complex mixture of multiple mechanisms. And I'll show you how that works. This here on the left is a measure of presynaptic neuronal activity, and it's percent change from control. So no effect would be no bar, okay? And you can see that it causes hyperactivity. The nerves are firing much faster than they should. Now, ordinarily, your brain and your wiring work that if I stimulate the neuron too much, the postsynaptic cell is going to desensitize. It's going to shut off its receptors and say, don't stop talking to me so loud, shut off, all right? That's not what happens with this. The receptors are upregulated, exactly the opposite of what happens in the adult. Nerves firing like mad, receptors are increased. So that should mean everything is hyperactive, right? No, it's deficient. Here's two serotonergic activity-related behaviors of many that we've studied. This one is the plus maze. I like this one because it's like walking the plank. You got two arms that are crossed and you suspend them off the floor, and one arm has closed walls and the other arm is open. So no one in his right mind is going to walk out on the plank and look down. Actually, females will do that. They're either braver or stupid. I don't know. No, they're smarter in other ones. This is just one of those sexually dimorphic behaviors. Now, here's males. They're cowards. Is that better?
Yes.
The males are cowards. They won't spend time in the open arms. But if they've had prenatal chlorpyrifos exposure, then their activity pattern is feminized. They become braver, or they're more impulsive. I see. This is what I love about behavior. You can shade the meaning of the result any way you like. Behavior is screwed up, and it's a sexually specific effect. Here's one where both sexes are affected, chocolate milk preference. Why chocolate milk preference? Because the people who study depression in models and animals have a set of behaviors that they use for a depression battery. And depression is most closely related to serotonergic dysfunction, particularly deficits in serotonin function, which is why Prozac, which increases serotonin function, works, right? So what you do is you take rats and you take their drink away, and then you give them a choice of water or chocolate milk. Now, nobody in his right mind prefers water over chocolate milk, right? And rats are the same way. The preference ratio is like 5-fold more chocolate milk. But notice that the guys who've had the prenatal chlorpyrifos don't show the same preference. And they actually, if you put them through the whole behavioral battery, they act depressed. They would be positive for someone evaluating an animal model of depression. So actually, their serotonergic function is deficient, even though the presynaptic terminal is hyperactive and the postsynaptic one is upregulated. So how does that happen? It's not innervating the right neuron. So you've got hyperactivity because the organism is desperately trying to somehow restore serotonergic function, but the wiring is wrong, and we've established that now through anatomical measures. Now, here's another example of miswiring, and one in which serotonin goes and replaces a function that should be cholinergic, acetylcholine. This is one of those hippocampal visuospatial tasks in the radial arm maze. And what we do is we let the animals learn stuff. And the Chlorpyrifos is subtle, they learn. And then we challenge them with a serotonin receptor blocking agent. Now, this is ordinarily a behavior that's governed by acetylcholine, not serotonin. So you do this to a control rat, here's no blocker, and here's increasing dose of blocker, and they don't care. They don't make any more errors. But here's a Chlorpyrifos exposed rat, early neonatal Chlorpyrifos exposure. They learn the task, and then it looks like the task now is being taken over by serotonin, because if I give them a serotonin blocking agent, they make mistakes. So here's a behavior that was supposed to be run by acetylcholine and instead has been miswired and is now taken over by serotonin. Let's switch to terbutaline, and I'm going to show you the same kinds of outcomes. Why terbutaline? It's a beta-adrenergic receptor agonist, so it's neuroactive, right? And we were using it actually not because of any interest at the time in autism, but because we were interested in how beta-adrenergic receptors control the fate of their target cells. And terbutaline arrests uterine contractions because it stimulates the beta receptors in uterine smooth muscle. It crosses the placenta and stimulates beta-adrenergic receptors in the fetus, which is why we were using it. And the arrest of uterine contractions means that it can be used for about 48 hours for a woman who's in preterm labor. It's not for maintenance use. It's actually contraindicated. It doesn't work. But as you'll see, what it does do is to cause damage and increase the incidence in humans of autism. The recognition of maintenance terbutaline tocolysis was withdrawn over a decade ago, and yet it's still widely used for that purpose. We did a series of animal studies over a couple of decades showing how overstimulation of these receptors screws up brain development. You know, big deal. I already told you that if you overstimulate receptors, you're going to have a screwed up brain. And along midway through this, there started to be some epidemiological evidence that the children of women who were treated for maintenance with maintenance tocolysis with terbutaline were showing impaired school performance and increased incidence of psychiatric and learning disorders. And then I guess for us, the pivotal thing was that we actually went and did, as a biochemist, I absolutely abhor having to do morphology. It's so slow, it's so judgmental. Scintillation counters never lie to you. You can't blind a scintillation counter, but you have to blind people who are counting cells. In this particular study, we used terbutaline in a window corresponding to its use in tocolysis. And as I said, I'm not a morphologist, although I did stay at a Holiday Inn Express. Actually, it was Lowe's, wasn't it? And here's the monolayer of Purkinje cells along in the cerebellum, and you can see they're basically wiped out by the terbutaline. It's at 44%. And there are all kinds of other abnormalities of structure. And what happened here was I got a phone call from Andy Zimmerman at the Kennedy Krieger Institute in Baltimore, and he said, do you know something that looks just like what we're seeing in the autopsy material of brains of children with autism? And so Carlos Pardo also has been invoked earlier in here as the person who did neuroinflammation in those postmortem tissues. So Andy Zimmerman set up a collaboration between our group and Carlos's, where we gave him our brain samples without telling him what they were. And it turned out that they had exactly the same pattern of neuroinflammation and microglial activation in the same places to about the same extent as he was seeing in the autopsy material from children with autism. When we did the behavioral analysis of them, they were hyperreactive to novelty, aversive stimuli, and sensory input. They're kind of jangly. Some preliminary stuff of social interaction shows that those are impaired. They also have cardiovascular problems that are indicative of autonomic dysfunction that also resemble some of the aspects of autism. And they went on, based on what we were doing in animals, to do some studies that I can't comment on since I don't know anything about doing these kinds of clinical or epidemiological studies. So I'm just going to cite that they then looked at dizygotic twins, because after all that has a high incidence of preterm labor, dizygotic twins whose mothers received terbutaline tocolysis for longer than 2 weeks. And what they found was the relative risk of autism in both twins, both dizygotic twins, both of them autistic, was increased to 2 by the terbutaline treatment. If it was male twins with no other affected siblings, then the relative risk was 4.4, right? And here, I think, is sort of the crunch. There are beta-adrenergic receptor polymorphisms that will interfere with the ability of the receptor to desensitize, right, which would then augment the effect of terbutaline. And for those individuals who have those polymorphisms, that they show intensification of these effects. So there's like your environment-gene interaction, except that it's a specific drug with a specific mechanism. But how specific is it? When we look at what terbutaline, a beta receptor agonist, does, for example, to serotonin systems, it looks a lot like what chlorpyrifos, an organophosphate pesticide, does. And they don't share the same mechanism at all right? So here's the effects of terbutaline on serotonergic presynaptic activity. They're hyperactive. There are some differences in the regional pattern, but it's still there. And here's the receptor upregulation from terbutaline treatment. The same exact outcome in terms of synaptic function, in terms of structure, in terms of behavior, from compounds that bear no chemical relationship to each other and no mechanistic relationship to each other. As a matter of fact, we then did something that sounds a little wacky, which is probably why I can never get it funded, right? We started, actually, you can have things that aren't wacky and they won't get funded anyway. It's my new motto for science is publish and perish. So what we did here was to ask the following question, translating rats into humans. And you do want to translate rats into humans and vice versa in an election year. So what if one of those sensitizing events that makes you take a bigger hit from an environmental toxicant isn't necessarily just your genetic background, but your chemical background? And what if it's a chemical background whose origins you could actually track, like preterm labor treated with terbutaline? So we did the experiment where we treat the animals as though it were treatment for preterm labor for terbutaline. And then we treat them postnatally with chlorpyrifos, but outside the window where the chlorpyrifos effect should be declining. So you're going to see a smaller effect of chlorpyrifos. We didn't want to hit them both in the prime vulnerable period, or you just, you max everything out. You need to leave some headroom for the interaction. So here's and this is just one of the measures, serotonin. Here's terbutaline. Here's that chlorpyrifos, and it's a little outside the window, so the effects aren't quite as nasty, all right? And here's the two together. And I'll call your attention to the brainstem. Little effect, medium effect, ooh, big effect. So what we've done is, with a drug exposure, unrelated to the subsequent chemical in terms of mechanism or type of compound, we've sensitized the developing organism to the second environmental hit. What does that mean? Well, I think this is sort of a recapitulation of the questions that I asked, only stated as declarative sentences, since I'm supposed to have proven something. So I think, since we're awash in this sea of neuroactive chemicals, I don't think there's any question that developmental neurotoxicants are likely to play an important, maybe even pivotal role in the increased incidence of childhood behavioral disorders, including ASD. And I'm not distinguishing ASD from the others in that regard. I think these are common outcomes, and the manifestation of it is a function of other things, other chemical exposures, genetic and environment, the plasticity of your brain and what it does with that insult and how it rewires. Disparate mechanisms and effects converge on common final pathways. So different agents could produce similar outcomes, and different agents that you wouldn't suspect to be interactive or additive can, in fact, sum up to give you worse outcomes. The sea of chemicals we're awash in is not one by one. It's mixtures and sequential. So I think we have a framework for understanding how the incidence increases because the complexity of the chemical bath is increasing. And what it also means is that mechanisms might not be the way to go. Convergence on common final pathways are probably the ways to identify these things. Lasting effects occur only when exposure is in a critical period. Thank goodness. Otherwise, we'd probably be in a lot worse shape than we are. And I've given you two specific examples that are relevant to ASD, one of which involves ubiquitous exposure. Basically, everyone's running around with organophosphate residues in their bodies. And one of which has a very identifiable subpopulation. And I'd like to see someone look at the interaction between these, because that can be translated into human populations. So, at the bottom line here, I've got what I think are the two main messages of this final talk of the symposium, that neurodevelopmental disorders don't have a cause. They have causes. Even if it's just one disorder, it has causes. And to try to say, oh, it's mercury in vaccines, or it's this or this gene, I think that's a waste of time. All of these things contribute, but there's not one cause that we're going to find for any of this. And so, and the other thing is that the origins of autism and ASD may not be so distinct from those of the other neurodevelopmental disorders. The manifestation, the symptoms might be distinct. But if we're seeking for causes and what chemicals we have to watch out for, we're probably looking at a collective bunch. Thank you.